All right. So today I'm going to talk about some analysis that Bedoy Fan and my group did on improving fine mapping. And it actually started off as trying to, let's say, identify what was happening with something that didn't look quite right in the data. And I'll get into that. All right, so I'm only going to spend a little bit of time talking about fine mapping, but one of the genome-wide association studies we were looking at is this one for alcohol and tobacco use. And this is one of the Manhattan plots for smoking initiation. And you can see a lot of parts of the genome are implicated, a lot of neural genes are in there, a very highly powered study. Well, if you go into some of these individual loci, there are some like this where you don't know exactly which SNP is causal. But of course, if you're in the field, you know that it gets worse. There are a lot of loci that look like this, where there are multiple that could potentially be the causal SNP. There are others where it tags more of a haplotype block, in this case, not even overlapping a gene body. And then there are other cases like this, where there's a relatively broad haplotype block actually overlapping multiple genes, and identifying which SNP or mutation is causal is a really important problem. And the idea is that there's a particular lead SNP that's picked up by the array, but there might be a functional SNP that's an LD with it. And we want to use various tools and techniques to identify what that functional SNP could be, including sets of annotations. All right, so one way that people do this is using evolutionary conservation, and that's where I'll start. And then I also mentioned that we compare the genome-wide association summary statistics to the epigenetic annotations using stratified LD score regression, a very standard technique in the field. All right, so our evolutionary annotations come from the Xenomia Consortium, and we're a part of the consortium. Irene Caplow, a postdoc in my group, will talk more about the evolutionary implications there. But I'll talk now about how we can use this for SNP fine mapping and disease interpretation. But just to touch on it briefly, as a consortium, we sequenced the genomes of 242 different placental mammals across many different parts of the tree. Now, often what's used to, let's say, identify whether a given SNP is conserved or not, or how it's evolved, is actually various models that are more or less all based on this Jukes-Cantor model from 1969, looking at whether or not a given mutation is more conserved than you would expect by chance if it has a certain a mutation rate or frequency across evolutionary distance. And of course, the implication is that if something is more evolutionarily conserved than you would expect or than the null model, then it's more likely to be functional. And you can kind of see this with scores like PILO-P and FastCons, which are much more modern than the Jukes-Cantor model. And here, for example, this is a protein coding region, and you can see that the wobble position in the amino acid code is under less selective constraint than the first two positions of the codon. All right, so evolutionary conservation is helpful, and I'll go in a minute or two into some of the numbers. But as people in the fine mapping field know, all the rage right now is cell type specific epigenomic annotations. And so I'll talk about some work that we've done there. All right, so we're basically using a TAC-seq for this. And so it's a proxy for, it's open chromatin, which is a proxy for regulatory element activity. And these regions of open chromatin are highly cell type specific, giving you power to not just fine map the SNP, but to identify which cell type or tissue is most likely to be involved. And so we're basically involved in actually building new cell type specific maps, in this case, of a brain region called the striatum that's really important for decision making, and has also been heavily implicated in neurological and psychiatric disorders like schizophrenia and addiction. And so we started off by basically building single cell profiles, even characterizing new cell types in the non-human primate striatum. And then we moved to creating epigenetic profiles with the TAC-seq of these individual populations. And so then we thought, okay, let's use the epigenetic annotations, a TAC-seq, single nucleus TAC-seq, and conservation to try and interpret human GWAS. And I'll say that what we observed is that when we just in general take a whole slew of genome-wide association studies and try to interpret them with different techniques, that actually we in the consortium found that some of the evolutionary conservation metrics like fast-cons in primates and phylop across all placental mammals were actually more informative than some of the epigenetic annotations like ENCODE 3 or fine-mapped EQTLs from GTEx. And the xenomia annotations are able to improve the power of fine mapping even more. But of course, it also depends on the cell type we use. So when we looked at these neuron subtypes of the striatum of the brain, they didn't really show an heritability enrichment for traits like snoring or bone mineral density or height that don't really have a strong neural basis. But the phylop score and the conserved SNPs was able to help us interpret and to fine map, again, based on the heritability enrichment. All right, so we started to see some strange things in the data when we looked at things like schizophrenia, so complex neurological and psychiatric disorders. And so here for schizophrenia, which is an extremely highly powered genome-wide association study, we didn't see much of an enrichment in the phylop conserved regions. And it's actually been observed before that evolutionary conservation doesn't provide a ton of information in schizophrenia. In fact, it even led to some speculation that schizophrenia relates to uniquely human evolution, although we also don't really see an enrichment in the accelerated regions either. And so how about some other complex neural traits like smoking cessation? So we also didn't really see a strong or significant signal of conserved SNPs for this trait, even though it's also very highly powered across over 1 million individuals. Audit score, which relates to problematic alcohol use, also a highly powered genome-wide association study, also not a lot of conservation here. And as running a lab that focuses on the genetic basis of complex neural traits, it was hard to reconcile basically these phylop or conservation metrics working really well for so many different traits, but then telling us very little about genetic variation underlying these neural traits. And so then we went on to epigenetic annotations. And as you might imagine, we actually got much better signal there. So for example, D2 medium spiny neurons, a particular neuron subtype, we saw high enrichment in open chromatin that's specific to that cell subtype. We also saw relatively strong enrichment of audit score problematic drinking in these D1 medium spiny neurons. And we started to put together maps linking specific neuron subtypes to these different traits. But we couldn't really let the conservation angle go. And so we were motivated by papers like this, showing that you can actually have conservation of function of regulatory elements in the genome, even if you don't necessarily have conservation of individual nucleotides. In this one paper, for example, they found that there's this enhancer scaper that regulates the gene islet. And basically, if you look at the orthologous region across sponge, zebrafish, human, and mouse, you actually see similar cell type specific patterns of enhancer activity in reporter assays, even though there's 700 million years of independent evolution and relatively low per nucleotide conservation. But what they did find is basically that similar combinations of transcription factors were present, even if you look, for example, between sponge and human. So that led us to basically try and marry the two, to marry evolutionary conservation with these cell type specific annotations to improve the ability of fine mapping. And we did that using convolutional neural networks. And so the idea is we wanted to learn the regulatory code that was present in each of these different specific neuron subtypes. And Irene Kaplow in my group, who, as I mentioned, is speaking next and was leading the effort. But some others in my lab also worked really hard to be able to build these neural network models that could take information in one species and reliably predict open chromatin in other species. And so what this looks like from an experimental design perspective is we basically train these neural networks on information from a handful of species that we collected data for, in some cases in our lab, in some cases from collaborators. but then we make predictions across all of the orthologous regions for which we have a genome and orthologs for. So another way of visualizing that is we basically start off with this matrix here up top, where the columns are the different species and the rows are the different regions of open chromatin. And we take the information from a small number of species, but then infer across this entire matrix of species by orthologous open chromatin regions. And so when we look at some individual sites, we can actually see results that make sense. So for example, there are individual mutations that have been implicated in traits like educational attainment or schizophrenia and cigarettes per day. And when we look at some of these implicated mutations, we can actually see that many of them tend to be conserved, not just in primates, but the orthologous region tends to be conserved in its predicted open chromatin, but for that specific cell subtype. But of course, it's easy to find these individual examples, so we wanted to look more systematically. And so what we did is basically stratified these based on whether or not the orthologous regions were predicted open chromatin across a broader set of mammals. And what we found is that the further you go in terms of evolutionary distance, the stronger enrichment you see. So for example, in relating smoking cessation to these D2 medium spiny neuron subtypes, we found the strongest enrichment in cases where we were looking all the way in bats, and whether or not the orthologous region was predicted to be open chromatin in that specific cell subtype. And we also broke it down into these different quartiles, and we found that the most conserved regions tended to be the ones that contain the associated mutations. We also found something very similar for audit score problematic drinking and these D1 medium spiny neurons. Notably, when we took other cell types that, let's say, haven't been as strongly implicated in these traits, like audit score and oligodendrocytes, which is a glial cell in the brain, we actually saw the opposite trend, that even though there was some heritability enrichment in human, that enrichment actually seemed to drop off the further you went in evolutionary distance, meaning that you could still map the orthologs of these regions, but they didn't tend to be predicted to be open chromatin in that cell subtype. So we actually built an entire set of maps from different neural cell types and different traits. We found an association between Alzheimer's and microglia, which makes a lot of sense, and also between different neuron subtypes and traits like smoking initiation. And in some cases, we were able to improve fine mapping by threefold using this tool. And this is just summarizing into various classes of traits. So we're able to find an improvement for D1 and D2 medium spiny neurons for substance use traits and to some extent psychiatric disorders. And then also look at the role of microglia in various neurodegeneration traits. We also see similar cell type specific patterns of conservation for EQTLs, but in the opposite direction, where the SNPs that tend to impact cell type specific gene expression, they tend to fall in regions of open chromatin that don't tend to be conserved, but are still active in that cell subtype. All right, so just to summarize, we combined information from evolutionary conservation from the Zoonomia project and these cell type specific annotations through machine learning to build an improved tool for fine mapping SNPs in a way that actually takes the cell type specific open chromatin into context and not just the individual mutations. All right, and again, it was really Bedoy Pfenning in my group that's leading this effort, along with Chai Srinivasan, another PhD student in the group, and Irene, who you will hear from shortly, built the models for cross-species open chromatin. Thanks a lot.
It's on. Hi, very interesting talk. What fraction of the SNP or variant in cell type specific regulatory elements are positively predicted by this cell test set? Based on the large standard deviation from the enrichment, I suspect that only a small proportion of the SNPs are positively predicted by your method. And also, I mean, it's good to see that you increase the enrichment, but how many SNPs do you think are false negative, basically, by applying the cell assay?
Yeah, so you bring up a really good point. So the error bars increase, and that's the general trend that we see. So the further we go in terms of evolutionary distance, the stronger the heritability enrichment gets, but at each stage we also lose some SNPs that are likely to be real. It's hard to estimate the exact proportion, right? Because for each locus, we have a whole handful of SNPs that could potentially be causal. But I would say, like a rule of thumb, and again, this is just generalizing from a couple of examples, we've created plots that look at the proportion of heritability explained by the annotation. And sometimes that proportion can drop by even a half across longer evolutionary distances, and yet the heritability enrichment itself will increase because that curve drops faster than the curve drops slower than basically the ability to map across species.
Okay, thanks.
Can you clarify? I mean, you were originally motivating this based on the fine mapping of like these genome-wide significant locations, but then it seemed your quantitative results were all based on the heritability enrichment. Do you know if these like improved heritability enrichments actually improve fine mapping, and do you have a way to quantify that more directly?
Yeah, that's a really good question. So in the end, we ended up using heritability enrichment, as our criteria for how well our annotation was doing relative to the others. For most of these loci, especially for something like the addiction traits like smoking cessation or initiation, we don't actually know the causal SNP, so it's hard to find a true positive. So what we're working on right now is basically using in vivo high throughput reporter assays in this part of the brain in the striatum to look at whether or not the SNPs that we find map with this technique can actually influence gene regulation in the specific cell types and tissues that we predict.